Hello, everyone. Welcome to Medicinal Express webinar series. My name is Ian Wan. We're really happy to have you with us for another exciting talk today. We have a distinguished speakers with us here today, Professor John Spencer. He's the head of medicinal chemistry at Sussex Drug Discovery Centres. He also has over 10 years of hands-on experience as a medicinal chemist. He has worked on several high-profile projects, including leading a synthesis groups for Nobel laureate Sir James Black's team. Professor Spencer's expertise across synthetic organics chemistries, drug discoveries, and Palladium catalysis, as well as cancer research. With more than 119 peer-reviewed papers, 13 patents, and over 9,000 citations, Professor Spencer's work has significantly influenced the fields of drug discoveries and medicinal chemistry. And his research has received numerous external fundings, and he holds an edge index of 44, making him as the leaders of his field. And today's webinars will focus on his latest research efforts, including the novel approach to fragment-based drug discoveries and the challenges and breakthroughs in developing p53 targeting molecules. And I'm quite confident that these sessions will provide valuable insights into the future of precision medicines and drug discoveries. So without further ado, please join me in welcoming Professor John Spencer.
Okay. Thank you very much for the kind introduction. If I can just share my screen, I think you've passed that over to me, so I can. Is this the one? Let's just have a look. Let's just see this. Is this the one? Can you see this all right?
Yes.
Let's just, is it advancing?
Yes. Could you make the screen yet?
Please interrupt if there's any problems. Right. It's a great pleasure, great honor to be invited today. Thank you. MCE and Sussex Drug Discovery, through one of my research fellows, Shamin Chowdhury, have had a really, and through Jeff from MCE, that we've had a really fruitful collaboration. He's sent me a publication actually where we've screened one of your libraries, and we've got some really interesting results that we'll hopefully publish soon. So it's been very valuable for having, you know, your compound collections. It's always good to have these in a suitable format for medicinal chemistry. You save so much time. Thank you for the invitation. I'm a medicinal chemist. I was trained as a palladium chemist, and I like to dabble in a bit of organic chemistry as well. So you see a lot of kind of catalysis, a lot of fragment-based drug discovery, and a lot of structurally led medicinal chemistry, because we're very well kitted at Sussex now with brand new X-ray, we're getting brand new NMR, and we've got really nice world-leading facilities to do medicinal chemistry and also biology in drug discovery. That's thanks to Sussex and thanks to some of the funders that I'm going to tell you about later. So my talk is really into three sections. We do some fragment-based drug discovery. We make fragments or small molecule entities. Fragments tend to have a molecular weight, usually actually below 250. You can go up to about 300, and you've got scope then to almost double the mass, the molecule, to still be within traditional Lipinski space of 500. And fragments fit nicely into pockets. You can find new druggable pockets, and you can have a fragment that it's quite promiscuous, that hits several proteins, but depending on the size, the shape of the protein, the environment, you can build it up through various vectors. And from one fragment, almost from one seed, you can create 5 or 6 different trees, if you like, different forests from this. So fragment-based drug discovery is something really that's come to the fore over the last 20, 30 years. There's several drugs on the market. And when I was funnily enough, when I was a medicinal chemist, we used to discard molecules that were micromolar, millimolar potent, but now it's because we can use very sensitive techniques, ITC, thermal shifts, X-ray crystallography, NMR, we can use this methodology to really detect small changes and small binding events, and use the excellent ligand efficiency that these molecules achieve to build up a drug and maintain that ligand efficiency. So I'm going to give you some examples of some of the chemistry that we've done, first of all, and then I'll move on to P53, which has really been one of my main preoccupations in my career over the last 10 years. Funded with worldwide cancer research, working with the first group at Cambridge, with mainly Andres Yerger, who's now at the SGC in Frankfurt. It really was a game changer, really, in my career. And I think we did some quite impactful chemistry and biology. So I'll move to that later. So this is the Hansch reaction. Hopefully we're on that on the screen. We're moving forward okay. And this is a traditional synthesis over 100 years old of thiazoles with this sulfur and nitrogen in this 5-membered ring. And basically nucleophilic sulfur will kick off typically a halogen leaving group like an iodide or bromide, and then you'll get this condensation. The entropy will drive that to form a nice 5-membered ring. This is brilliant. This is used a lot in organic synthesis, but it's quite, let's just say from an environmental point of view, it could do with some improvement. You're giving away HX, so you're giving away acid. If you're doing this to scale, you've got acid to get rid of, you've got to neutralize it. You have a problem neutralizing it because of the thermodynamics, get too hot. And maybe some of the groups on the thiazole, if you've got a bot group on there, maybe you'll start deprotecting it when you're doing it. And you've got to make these bromo, let's just get my pointer on. There's the laser pointer. You've got this X group here that's leaving. Well, these can be alkylating agents. They can be lacrimatory. They can cause, irritation. And not all this chemistry is compatible. You can't always have the right R groups here. So I'm very proud to have a collaborator, Storm Hassel-Hart. He's now a lecturer at Sussex, and this is really all of his work towards the end of his postdoc that he did. We had an EPSERC-funded postdoc for 3 years. He came to my group, went to UCL to work with Jim Anderson, and now we're really happy to have him back as a lecturer because he's really the future, one of the few people that no pressure, but he's the future of Sussex organic chemistry. And he devised this method using iridium catalysis, and these are the compounds he made. He was really fabulous at making arrays, making 5 or 6 compounds at a time and doing quick purifications. And this represents just a couple of months of his work. I mean, it's a whole PhD's work here, really. And he's made these sulfoxonium ilids that are alternatives to diazos, so they're much more safer, easy to handle, easy to make. And what you do with the iridium is they've got a negative kind of zwitteronic nature, negative carbon, positive sulfur. The negative anion can kick off a chloride on the iridium. The iridium can kick that back, kick out DMSO, and you've got a C double bond iridium complex, a carbene, which can then react with a sulfur, and then it can come round and cyclize. So we're not using a bromide here or an iodide as a leaving group. We're not having to make these alpha bromo ketones or alpha bromo carbonyl compounds. And you can see it's quite the scope is quite good, actually. Phenestizole, we always like to, when we do this chemistry, show real world application. So this is a drug for inflammation. We can make it in a simple step. So it's quite a convenient catalytic route to this particular molecule. It's got really good substrate scope. So we wanted to make these molecules fairly diverse. We didn't use any programs. We didn't use any kind of statistical or data analysis. But you can see you can have hydrophobic groups. It depends on, if you're making these compounds to bind to a particular protein, for example, they might be sticking out the protein in this area, so you might need a solubility handle. And a morpholine there, there's a fluorine there, which is useful in medicinal chemistry. So it's this is an escape from flatland molecule with an sp3 carbon here on this tetrahydrofuran. So it's quite broad in its substrate scope, to be honest. And we've taken it further. Storm made these simpler thiazoles. Thiazoles, aminothiazoles, can be problematic in liver hepatotoxicity. So they can make thioureas or compounds that can interfere with glutathione, and you can have a hepatotoxicity alert. Other ways of making thiazoles without that amino junction are these particular molecules. You see, it's quite broad with heterocycles. The Hunt reaction is not always so broad with pyridines, as far as I'm aware. So we can get pyrimidines and pyridines in there, different positional isomers as well. And the yields are fairly good. These are not really optimized yields. These are reactions that Storm carried out really without really putting in months of optimization. So it's quite a general and might be an adopted route. We're getting quite a lot of citations for this paper. Quite a nice route to making these particular molecules. And you can even make selenozoles that are much rarer than thiazoles. It could be a nice bioisosteric change. It could be a patent expansion. You've got some thiazoles with activity. So maybe you want to take an example of one of your molecules and make the selenium so a competitor doesn't come and patent bust your molecule. Or get around your patent. So this could be useful, and maybe it's got different types of reactivity. So it's nice to know that this was also amenable to seleno heterocycles as well. So that was quite nice. This is kind of back to front the way we did it, but the way to present it is much more adaptable. We started this project because we got a crystallography hit from a fragment screen. Our dear old friend Stephen Hare is a wonderful, wonderful crystallographer, a lovely colleague. He unfortunately died in an accident halfway through this project. It was really, really, really tough on his family. It was really, really tragic. But this part of the talk is dedicated to Stephen's memory because he's still in our heart, still in our memory. And he got some fragment hits, and one of these thiazoles came up as one of the hits where we thought, let's just elaborate, let's remake it, let's try and set up an assay and look, see if we can work on it in this kind of resistant gonorrhea antimicrobial research project, because we've got a problem in clock kicking in 25 years time. A lot of our antibiotics are going to stop working. We're going to have 10 million deaths a year in the world. It's a pandemic that doesn't pass like a COVID pandemic. It's there for good. You know, childbirth, garden accidents can be really, really problematic if you can't access the correct penicillins or the cephalosporins of the future, or the other compounds. So we aim to kind of build upon these fragments to try and bring in activity, try and bring it in, but Storm found it kind of difficult to make these kind of compounds by hand chemistry. Hence, back to drawing board, let's make them by a new route, let's make it into a synthetic program. And this is quite long-winded, but we've got the iridium catalysis, we've got the thiazole, we've got a reduction with a nitro group, obviously avoiding acidic conditions because you've got a bot group. So this was just a simple palladium on carbon with hydrogen. Take off the bot group and then do some other chemistry, do some and in this particular case here, obviously because of the similar reactivity, the aniline and this aminothiazole, do an amide coupling first. It's quite general, it's quite easy in scope. It's just a simple amide coupling reaction, take off the bot group, and then we've got these molecules. And as we can see, one of these compounds was a hit in this particular screen. So really, this whole project was based around this particular biology. For obvious reasons, for very tragic reasons, we weren't able to take this forward. We are taking it forward now. We are working with the INEOS Centre, and we've had some interest from industry. So, you know, Stephen's legacy will be on this project, and we'll make sure that we remember him and we see it fit to see his science through. So he was taken away from us way too early. So that's an example of some of our fragment work. Our work is very, very collaborative. I was a keen football player, I played in goal. And you learn that a football team's only good when you've got good players in front of you, when you've got everyone, you've got your defence, your midfield, and your attackers. And I, as a goalkeeper, I'm always lining up my defence and shouting at them. I don't shout at my team, but it's good to have the right team around. And I always think that, you know, if we try to set up assays as chemists and try to do molecular modelling and crystallography, we'd be spending a lot of time making a lot of mistakes. Why don't we do what we're good at? And why don't we work people who are good at what they're doing, who can't make the molecules, bring it all together, and you get a perfect synergy. You get a 1 and 1 equals 3, a perfect synergy. So this has worked with the Kostakis group at Sussex. They've been doing some fabulous multi-component reactions. And they've worked on this A3 reaction. This reaction's been around for quite a while, but they've been looking at ways of doing this a little bit more atom economically, and also using, for example, modern green technologies such as solventless ball mill grinding reactions where you don't have a solvent. So obviously you're not using chlorinated solvents, for example. So this reaction is great. You've got a copper catalyst. You've got an aldehyde that reacts with an amine to form an imine, or sometimes an iminium. The copper comes along. It's almost like a Sonegashira or a Chan-Lam type reaction. You've got a copper acetylide that's formed, and that will attack the imine, and you form this carbon-carbon bond. And you've brought together three components, a little bit like an Ugi reaction. It's a multi-component reaction. And you've created molecular kind of complexity simplistically by templating it together with copper. You can do this chirally. We've chosen not to do it chirally, and I'll explain later why. By chirally, I mean we've not made molecules with 99% E as the R or as the S. We tend to make the racemate, but I'll explain that later. And George led this project with our wonderful PhD student, Jack Devonport, who graduated a few years ago. He's got about 6 papers from his PhD. We've got another one. We're just waiting for some IP to be settled and some other results, and we'll get that out soon. He's now in Mexico, I believe, teaching chemistry. He's a wonderful teacher. And we chipped in with some examples. I always think that once, as I said, with the thiazole chemistry, it's always good to have a real world example to show that this is not limited to what we could call methyl, ethyl, butyl, futyl. We can have examples that, you could confidently think, oh, I'd love to put this group on the end of my drug to make it more soluble, maybe to have a solubility handle, maybe put on a PROTAC handle or a linker for antibody drug conjugation, or maybe just to improve the solubility of the molecule because it's no longer flat, take away that flatness. And we took JQ1, for example, a very, very famous BRD4 inhibitor. And the alkyne part of that, the CH of that, this molecule is very easy to make, this propargyl amine. And then you can just simply link it onto here and have the R being the JQ1 fragment here, or a steroid-like fragment, or a ilotinib, a kinase inhibitor. It's not the best part to put this from a medicinal point of view. This is in the hydrophobic kind of gatekeeper residue. I doubt that would actually bind there. It will probably stop binding. You really want that group here, but we're just showing from a synthetic point of view, you can do late stage CH activation, late stage functionalization. And this was really the cherry on the cake in this study because, we were very, very happy to be able to do this with some, drug-like molecules that really, might be useful one day. We've since taken this chemistry forward, I believe. We, as I showed before, we had an alkyne with a JQ1 on here, or a ilotinib, or a steroid, and did some chemistry. And we thought, well, why don't we just leave that open? Why don't we do this chemistry where we can take an alkyne, we can take an aldehyde, it reacts, these two react to form an imine or an iminium. The copper makes a copper salt so it's like a CH activation of the alkyne that attacks, and you form these rather complex yet simple. So you use simple chemistry to make quite complex, tunable compounds. So we can change R1 to whatever we want, really. These heterocycles can be really whatever we want. These aldehydes can be really whatever we want. So the permutations are almost endless. And Andrew McGowan, a super research fellow in the group, has worked on this and really taken ownership of this. And we're very, very close to submitting a paper, very close to writing it up. I expect to have it ready for the end of the month. No pressure there. And these are some of the examples. And a few that I'd really like to bring to the fore are the ones with the halogens here. You've got the chloro, the bromo, the bromo, in many cases. And it's quite, I was very surprised. We had a group meeting where Andy said, I'm going to do this. And I wanted to stop him and say, it won't work with a halogen. It's going to interfere. It's going to oxidatively add with a copper. And I let my group do things. I love being proven wrong. And I think this was great. And I think this was the beauty of science. If I'd have been in control of this, there'd be no 5B, there'd be no 5D, there'd be no 5G, there'd be no 5J, there'd be no 5H. So please go and do those reactions. Don't listen to the supervisor, is one of the take-home messages. And I think that's great. It's great to have a group that feel, I guess, that you've got the freedom to try these as long as it's safe. You know, don't do something dangerous. But sometimes do it. What I think and what we think on paper doesn't always happen in reality. So we've made all these compounds. I said we've got a chiral center in the middle. We've got this amine moiety. We can have a hydrophobic group here, an oxygen, a halogen here. So you can have a hydrophobic group there. You can have a hydrophilic group. And it depends what you make. You might want to make these and, don't know, make some drugs for them, but you might want to link more than one drug. And this bit might be crucial. This part might be sticking out into solvent outside the pocket of the drug. This might be still inside the drug in a hydrophobic cavity. That might be too big. So you might want a smaller isopropyl. So we've done this really just to show synthetic kind of utility in this chemistry. I don't think I've added the latest results from Yan Lin. We've got Yan in my group, who's a wonderful Royal Society KC1 fellow who joined mine and George's group, Kostakis's group, at the start of the year, and she's done some really nice chemistry with orthogonal protection. And I'll leave that as really as I won't give you a spoiler alert with that. But then you've got 3 different orthogonal handles on your molecule. You can link on your first amine, for example, with a coupling, protecting group on the second aldehyde from the aldehyde, which I've not shown here. Yeah, well, we've got a bot group here, we've got a bot group there. Imagine we've got a bot group here, we've got maybe an FMOT group here. You can take the bot group off, do a coupling, an FMOT group here, do the coupling, and then you've got a bromide there that you can displace. You've got 3 different angles to automate this chemistry, to make this chemistry, to send these vectors off in different directions, to make different diverse compounds. And the fact that they're RS mixtures, you've got the diversity that you've got both the R and the S. You've doubled your money every time you make these molecules. And that's why we kept them as racemates, because they're quite, they're quite diverse as they are. And you don't know whether you want one enantiomer or not. You can get one enantiomer, maybe with an enantioselective synthesis, or you can do chiral purification, and I'll get onto that next. Now, how did we work out the structure of these molecules? We've done some 2D NMR, some Manvendra Sharma in NMR has done some wonderful 2D spectra for us to show us that. And I'm not the biggest fan of doing all these complicated NMRs. Sometimes X-ray crystallography just solves the problem for you. So in 3 or 4 of these cases, we've got X-ray crystallography that shows the connectivity. And this is thanks to the National Crystallography Service, Simon Coles and Graham Tizard there, who's fabulous friends and fabulous crystallographers. So they'll be on the paper as well when that comes out eventually when we submit it. So we've got a nice way of making simple building blocks, and we haven't stuck really a molecule on there. If we had JQ1 on all of these, this first molecule, there'd be BRD4 bromodomain actin molecules. That's great if you work on bromodomain actin molecules, but if you're working on, say, PROTACs, or you're working on hybrid molecules, and you've got a KRAS, or you've got a CMYC, or you've got a EGFR or P53 targeting drug, you can put that on. You can clip it on at the end if you've got a particular reactive group. Do a Mitsunobu, do a SN2 reaction, and you can put it on as long as you've got a handle on there. So this is why we keep this quite open. We can make chiral compounds. We can make these to scale. So Andrew McGowan has done this. I hope I said Andrew McGowan. I always mix him up with another colleague of mine. Andrew McGowan has made these on 5-gram scale, unoptimized in about 50–60% yield. And we've worked with colleagues at Reach Separations up in Nottingham for doing our separations. So you get a 1-to-1 mixture of these 2 enantiomers. They look different, but the area under curve is very similar. And they're able to readily separate these into two separate enantiomers, but we didn't know which one was which. So we had an intro via Ed Hoggart at Reach to US Biotools with Rena and with Jordan Nafi, Rena Decourt at Biotools, who are based in the US, and they use vibrational circular dichroism to give you the absolute configuration, even of an oil. So you don't need crystals of your molecule. You can actually just give it an oil. They run a spectrum. I try to simplify things that are very complicated, and I think of it as something like infrared with a polarimeter. And you can separate a left and a right-handed form of a molecule. One's up, the other's down, and you can do calculations to work out the spectrum of each enantiomer. And this is work done by Nafi Senior for very many years. And we covered many aspects of this. This work has not been published. This is part of the other story with the A3 chemistry, but an earlier aspect of the use of VCD and the very difficult kind of scenario that we've met in some really unwanted examples is when you buy some start materials that are not what they say they are on the pot. When you've bought the R and it's actually the S. When you bought the S, it's actually the R. When you've bought a 2,3 regioisomer, it's actually the 3,4. It can cause you lots and lots of problems because you might be repeating the synthesis of a drug-like molecule, putting it into animals, and you've inadvertently put the toxic enantiomer into the animals. It causes unneeded, unnecessary toxic side effects. So VCD like this would give you the absolute configuration. And a lot of companies need to do this before they can take something forward, even if it's a resynthesis to confirm that. So they might just put it on HPLC and get a retention time and show that, and they'll know the absolute configuration that actually matches this. So these two fractions were separated. One of these fractions, I can't remember which it was for this. It's the R. One of these was put in. It's probably the second fraction. No, the first fraction here. The R was determined by vibrational circular dichroism, which is a really, really super non-invasive technique. You need 5 or 10 mix, and you can get your absolute configuration of your molecule. We've used this, surprise, surprise, no surprise, to make heterobifunctional molecules such as Protac, and we've taken JQ1 because it's a good benchmark, lots and lots of early, and even Protacs now is still being made with this wonder molecule, really. So Andy McGowan took this broma compound, substituted it with this left-hand side of this JQ1 with this thiophenol moiety on it, made this molecule with a racemic group here, and then attached an E3 ligase ligand, so a CRBN or VAGL molecule here to make these heterobifunctional molecules. We had no kind of concept. We didn't measure their properties. We didn't predict their properties. We didn't look at any conditions. It was really just to showcase that this chemistry can be used, these linkers can be used in drug discovery programs. And we managed to set up a strategic partnership with the Chuli group, Alessia Chuli group. They've been great friends with Will Farnaby and Vesna Vetna there. And Andrew McGowan went up to Dundee for a month and did some of the tests. It really was so welcoming and great. And we're going to write this paper with them, of course. And one other compound that was made, this is done by Damien Kaipang in my group, wonderful postdoc with lots and lots of industrial experience. And I think this is really the future of these A3 reactions. And we just showed one example, but you can take an aldehyde in green. So this would be the aldehyde here. Take this amine. So I've showed in a way the mechanism, if you like, halfway through. We've got this iminium. We've made the copper complex of the alkyne. It attacks, and you form a racemic group here. We've got a chiral center. I think it's too far away to get any chiral induction. You've probably got an RS mixture here. And unfortunately, these two groups are not orthogonal. We can do chemistry, obviously, with orthogonal protecting groups and deprotect stepwise, but just showcasing the chemistry. take off both bot groups, put onto thalidomide type molecules. These are CRBN E3 ligase ligands, and these got tested. But this is what we call plug and play. It's very, very quick, very simple to operate, and really could be used in a kind of an open trans format to make these kind of compounds if you have the right connection. And obviously you can vary your aldehyde and your amine motives. You can vary your cerebrons and your VHLs and your linkers. We tend to find the VHLs are quite insoluble, so we often prefer the CRBN ligands. And just to show you this again, so that's Andy Andrews, then Damien's chemistry here. And we've got some PROTAC data. This particular compound 7A looks to be the most potent. It's taking out the protein. We can reverse that with a proteasome inhibitor, for example, a nedylation inhibitor, and we can do that in a dose-dependent manner. So they are work as PROTAC. 7B less so, it's quite insoluble. 7C, probably the linker length is too short for this. But again, we're not making PROTACs for the sake of making PROTACs. It wasn't a SAR, make 20, 30 PROTACs. We've done that in other programs, and you tend to need to make quite a significant number of PROTACs before you get activity. This was just really, look, what can we do with this A3 chemistry? How can we take it forward? So great thanks to Dundee, great thanks to the Kostakis group, to Jan, to Andy, and to Damien. And if I've forgotten someone, I do apologise. And Claire as well. Claire Adcock was very another excellent group leader. Was very good with some of the admitox and getting the admitox stage on some of these. They're not particularly impressive, to be honest. The clearance is quite high. Maybe the sulfur's a problem. I think we got some clearance data on this and some permeability and solubility, and it's not brilliant. It's not surprising, but as I said again, just to showcase the chemistry, use our chemistry, design your PROTACs better. We would just have to quick wins and to show application. So that's the kind of the fragment part and talking about the A3. And I'll move on and talk to you now about some P53 chemistry that I've been involved with for over 10 years with a very eminent chemist, Sir Alan First, FRS from Cambridge. One of his research fellows, Andreas Jürger, who's now moved on to SGC Frankfurt and still working in P53. So we've been working on, and lots of collaborators, Matt Bow, and other people in the first group, Riser, Ree in my group, Adam, ex-PhDs and postdocs in my group have done a lot of work in that. Y220, 220 tyrosine is found in wild-type P53. There's a mutation that's found in about 100,000 new cancers every year, and it's where the tyrosine, a large tyrosine with a kind of phenol group, is mutated to a CH2SH, a very small hydrophilic, nucleophilic sulfur group, and it creates a small crevice in the protein. So you got this little hole in the protein, and what happens really is, in the kind of simplest form, is that the protein no longer is thermally stable. So because of this single mutation that's cropping up, it's like a gain of function in P53. It's not hanging around. Its half-life is too short in a human, for example, or in a physiological context. And apoptosis, the role of apoptosis is lost. So the cancer has a gain of function. The cancer can evade apoptosis, one of the hallmarks of cancer, and can continue. So the first group many years ago studied this. They produced these crystal structures. They looked at the thermal stability of the protein, showed that this particular protein, I think it's about 8 degrees Kelvin, less stable than this particular protein in terms of its melting point. So at 37 degrees, centigrade in the body, there's not much of this around. It's going to be totally unfolded and it's going to degrade. So the idea was, because there's a little hole here, a little kind of cavity in the protein, can we put a molecule in there just to, it's almost like a door stop to stop that door slamming. Can we just hold it together? And if we do so, can we rescue the wild type behavior of the protein? Because if the protein's got a longer half-life, it can still read DNA and trigger apoptosis when needed. So this really pretty terrible depiction really is, there's your mutation. You've got your 220 cysteine instead of a tyrosine here. The protein just destabilizes and it unfolds and aggregates. If you can put in a molecular chaperone or a stabilizer to sit into that cavity where it's a mutation induced missense mutation, you can stabilize the protein in such a way that it's got a similar melting point to the wild type protein. It still goes about and reads DNA and triggers apoptosis. We can re-trigger apoptosis. That might synergize with the use of chemotherapy, for example, for patients with this Y220C mutation. So one of our first forays in this area was using fragments. This particular molecule came out of a screen that we were working on, and it wasn't particularly soluble. So it's at the time when I was still in the lab, and we realized that this nitrogen was probably solvent facing, or this alkyl group was. It's just a matter of putting on a solubilizing group that can be protonated and stick out, and we got this particular similar analog to this PK7088, and we got a crystal structure. It's always a good idea. It's always highly desirable, not always achievable, to have crystal structure evaluation, structural evaluation behind your study. There's the cysteine of the mutation at 220. You don't have a tyrosine there, and this is where it sits in that cavity. Can we move on for this? And this was one of the early studies showing that in these proteins, you've got a very small amount of correctly folded protein. You can use antibodies to look at correctly folded wild type protein, and this is unfolded protein that's going to denature. When we add this particular compound here, about 200 micromolar, it's not a very potent molecule, it's not ideal. You greatly increase the amount of correctly folded. So you're rescuing the folded wild type form of the protein. You're greatly reducing the amount of incorrectly. So it's aggregating less and it's unfolding less. And they did this is at Cambridge in the first group, and they had very little effect on other mutants. So it did seem like this was working on this particular mutant. It's not just a kind of a pains kind of panassay interference or some kind of off-target effect. It does seem quite linked to this. This is the paper. There's a lot of experiments that kind of pinpointed this PK7088 as a useful tool, although we need to get, a couple of orders of magnitude in potency to make this like a tool compound for P53 drug rescue, mutant rescue. So another series that the first group had worked on using in silico models was the development of this PK083, which is a carbazole. And we can see that the DNA binding surface of P53 is up here. This is where the mutants occur. So it's quite far away from the DNA binding surface. And they looked at this as Yerger again, Andreas Yerger from the crystal structure, showing that there's a key hydrogen bond between this NH methyl and a backbone carbonyl of the aspartic acid at 228, not the side group, the CO, the amide bond, the peptide bond are there. And this sits into this hydrophobic group that's got lots and lots of hydrophobicity. And there seems to be a solvent exposed area here, kind of a solvent exposed area over here. And there's the cysteine. Sometimes you get more than one population in your crystallography, and you can see more than one cysteine molecule. It's 50% of one, 50% of other, or 80-20, depending on the population. So this started off, this was a kind of blueprint to try and improve the affinity. We've got a subsite 2 that we call here, a subsite 3 that's a small pocket. We looked at SAR. You can have an ethyl group there, you can have a propyl, cyclopropyl there. You go bigger with a benzyl, you kill activity. And this is kind of open SAR. You can have lots of groups because they're solvent exposed. So lots and lots of amines with that NH is there. If you have the N-methyl-2, you lose that hydrogen bond, it flips backwards. That N-methyl-2 flips backwards and performs van der Waals. You get van der Waals interactions with this valine, and you lose a little bit of affinity. So that's interesting. So we've worked on this. We got 4 years funding from Worldwide Cancer Research that really took our research to the next level. I got my professorship probably on the back of this, thanks to WWCR, who've been really wonderful in this respect. We looked at optimizing this PK 083 using structural chemistry. And one of the ways we wanted to do this was to engage this cysteine. Can we actually alkylate this cysteine? We are quite close. And we tried a whole range. We couldn't make this molecule, but we tried a whole range of molecules. Can we make an SS bond if this is a prodrug? Can we open this up like a mustard? Can we directly alkylate even the iodo and bromine? And these just failed miserably. They didn't work. We have recently worked with a Gaydon group in Strasbourg, with Andreas Jürger again in Frankfurt, and Matt Bow in Southampton, and collaborators at Edinburgh. And we've been able to do Michael chemistry. So this is AG3. This is from our collaborators group. And we've been able to show that this acts as a Michael acceptor on lots and lots of cysteine. So this is a protein, you can alkylate it. It also opens up. And we were using this zinc binding moiety to see if it can work as a dual Michael acceptor and a zinc chelator. And we made a couple of controls. This can't be a Michael acceptor. This can't be a zinc chelator. And this was very reactive. This wasn't reactive at all. And we're using this as a way of trapping the cysteines in P53. So you can get covalent engagement of P53. This is not specific. It works on lots and lots of surface cysteines, not just on the Y220C, also on wild type, but it's a start where we can probably build in some selectivity. I sometimes have memory glips. Sorry, it's Amanda Jarvis at Edinburgh who gave us this. She's an excellent early career researcher at Edinburgh. And we're submitting that paper shortly for review. We weren't able to covalently engage the cysteine, so we thought of other ways, and we looked at halogens such as fluorine. Fluorine is ubiquitous in medicinal chemistry, blocks metabolism, it mediates, it picks up very weak electrostatics, very unlikely hydrogen bonds, more electrostatic interactions. It can modulate the pKa of a molecule, an amine. So it's not surprising, probably a quarter of all drugs have got one or more fluorines in them, so we've got several fluorines. So we looked at this PK083 and thought, well, if the cysteine's nearby, what if we monofluorinate it, difluorinate it, trifluorinate it? So we're not physically doing late-stage CH fluorine exchange. We were just taking the fluorostarter materials and putting them onto the central nitrogen here. It's not straightforward working with fluorine. It messes up the electronics. You get lots of unprolonged effects. You sometimes lose HF rather than a bromide. So we've had elimination of HF in these reactions. But what we did, we had the monofluoro, difluoro, trifluoro. Here's the monofluoro, and contrary to what we predicted, the fluoro doesn't interact with the cysteine. Again, you see two of these populations, as you often get in crystallography. And the fluoros interacted with the backbone carbonyl. When you've got two fluoros, you've got the backbone carbonyl again, but now it's picking up an SH. Three of them are picking up more interactions, and we're getting increased affinity with this trifluoro compared to the mono and difluoro. We've got the dimethyl compound there again. It flips backwards. We made that as a kind of control. And we're going from 150 micromolar fragment, quite weak, to a 30 micromolar molecule by having these fluoros. And Nick, meanwhile, did this did a beautiful job of this in a review on fluorine in medicinal chemistry. And you've got the HF interaction, also F into this sigma star, sigma hole in sulfur. That's quite important in fragment-based drug discovery and drug discovery. We got affinity by ITC, these beautiful curves. So fluoroethyl as opposed to ethyl can be very useful in picking up backbone carbonyl interactions and SHF interactions. We've published a lot of that. We did some calculations, albeit on a much more simplified system, and we thought the monofluoro would be the most potent and the trifluoro would be the least potent. We didn't take into account desolvation penalties that you get when fragments bind into hydrophobic pockets. So the monofluoro is quite polar. It's very difficult. You've got a very big cost for desolvation. The trifluoro has got very little desolvation. So the trifluoro has got good activity, no desolvation. The monofluoro has got good activity, but very high desolvation. That's why the trifluoro wins that kind of balance. And it's always the same in medicinal chemistry. It's never your best compound, your most potent, that wins the game. It's always the one that's got the best balance. It's always a kind of a balancing game. We've done other things with P53, again with Worldwide Cancer Research funding. This is again some beautiful work done by Andreas Jäger with this PK 083 start material, establishing a whole kind of pocket, this plasticity in the protein. We've got a beautiful structure paper. It's got lots and lots of very expensive color images in there that we published in color. And we exploited this subsite too. And you can brominate this particular carbazole or build it up using Cadogan chemistry and have this bromo here. And then I'm happy. We're doing palladium chemistry. I'm in my element. We're doing lots and lots of palladium couplings and picking up interactions in this particular area. And this summarizes really the Y220C protein that we've built upon from the first group, and we've built on it with Worldwide Cancer Research funding. And I know Andreas has got some Deutsche Foundation funding as well, some DFG Foundation money in Germany. And we made a lot of compounds. The bromo, already you've got a 10-fold increase in potency. How that bromo substituting can open up a hydrophobic pocket. It really, really is important sometimes. If you've got a hydrophobic pocket, stick a halogen, move it around a bit, just see if you can get some gains in affinity. And it's then also a handle to do some coupling chemistry. So we went a little wild and did a lot of coupling chemistry, gained some very important structural information from these molecules. And we found really that thiophene was really, really good. Only substituted in certain parts. We did a lot of regioisomer switching. We did a lot of methyl scans, and we're able to finally make single-digit micromolar compounds. Not quite in the nanomolar range. They're still quite small, but we made some quite good affinity gains doing this. These are crystal structures, excuse me, showing conserved binding of these molecules. The thiophene points towards the cysteine. I did think there might be a sigma star kind of SH to the empty sigma orbital, the non-bonding orbital of the thiophene, but we're not 100% sure of that with the crystallography. And obviously, crystallographic packing can have influences in the way these things bind. But we made a nice series of molecules from this. One thing we did find is that they can be quite cytotoxic at times. So you put them in cells, sometimes you don't see specificity towards Y220C. And we always see this in the mass spec. We always see loss of NH-methyl. And talking to a colleague, Mark Bagley, he suggested there was this type of molecule as an intermediate, this quinomethide-like molecule that might be cytotoxic. And it has problems. We have a few problems with HepG2 and H2 alpha gamma phosphorylation, which is a hallmark of DNA damage. So these molecules, taking them forward, there would be a few issues in trying to develop them as drugs. But it's been a very, very good academic exercise. They trigger apoptosis and pull down and kind of target genes such as P53 mediated, upregulated mediator of apoptosis and other regulators. So this was quite a useful tool compound for cell data. We've looked at other mutants. This is Andreas Jäger again, looking at Y220X, such as serine or asparagine, Y220X being S or N in that particular case, and that's quite nice. Very rare compared to Y220C, but still a pan stabilizer of these mutants would probably be useful in cancer. And some of the influence that I believe we've had in the field has been that this is a molecule from PMV Pharma down here. We made this, we weren't aware of their clinical candidate at the time, but I think they're in phase 2 with a P53 stabilizer, and you can see similarities between first group compound. It's almost like a merge of those two compounds. Take out this AR group, merge the two fragments, keep the blue, keep a little bit of the red, take out a ring, which is always good, an aromatic ring count in drug discovery, and then you've got this molecule. And Andreas has done some beautiful X-ray crystallography and shown some very good overlay between these molecules, which has been really nice. Showkat, a real icon, a real leading chemical biologist in the world, a really nice, very, very supportive in collaborations and very supportive in correspondences, made this molecule. I did say that we tried to make this molecule. They couldn't make the NH methyl, so they had CHO here. We never thought of that. And this is a covalent stabilizer of P53, which, you know, builds upon some of this work. And these are some ACE tags. You can see that the SAR in our work it was quite nice to know that the methyl group was important, that we'd shown as well on this thiophene, and they've put this together with a histone acetyltransferase ligand, and it can acetylate a key residue in P53, not only trigger apoptosis, but acetylate epigenetic modification of P53 at the same time. I think it's K382 that gets acetylated. So these are ACE groups from the group in the US, from the Yin group, I think. Finally, if I've got another couple of minutes, if I'm okay, just to go through some chemistry where we've worked on fragments and been looking at some unusual bromodomains, such as FIP2, which is a plextrin homology interacting protein second bromodomain. It's an atypical bromodomain, doesn't have an asparagine in its acetylated glycine reading pocket. It has a threonine, and you can get an extra water in there that we call the FIP water. And we started on a screen with the Paul Brennan's group, and they gave us lots and lots of crystal structures with Frank von Delft's group as well. And I wrote a grant on the back of this paper, which is on the poised library from Diamond, on the elaboration of these molecules, and we took this forward. So poised fragments are fragments that you can elaborate, you can get a hold of them, and you quickly turn them around into new fragments, do lots of quick automatable, if you like, and easy plug and play chemistry, using some of the toolbox of the medicinal chemistry that we have at our disposal: Suzuki, Chan Lam, sulfonamide, amide couplings, heterocyclization, you name it, the reductive aminations, that type of chemistry. We were finding it was quite slow at some stage to get from, you know, fragments to more drug-like molecules, because some of the chemistry is quite traditional. It's single reactions, lots and lots of extractions, lots of manual purifications, single evaporations, means that it becomes a very tiresome, cumbersome kind of chemistry with lots of bottlenecks. Can we cut that out? Can we cut down the exposure as well? Can we cut down the use of solvent, the interventions? What we did, and this is not a, please forgive me, this is not a advertisement for open transfers. I think there are other, Zinza do this, other manufacturers do really nice automated systems as well. But it's quite a cheap, you know, about £6,000, £7,000 unit, where you can do 96-well type chemistry. And Storm, when he was doing his postdoc with me, went up to Diamond to work with Anthony Amon in the Frank von Delft group at Diamond, which is a national synchrotron facility. And they sought to do much quicker fragment-based drug discovery, and to do crude reaction mixtures and X-ray crystallography without any intervention, really. So the idea was to use FIP as a protein, to generate some fragments against this particular protein, then to find a fragment where we thought we could go in different directions. As I said before, you go in these different kind of vectors, use simple chemistry that can be automatable on a small scale to do this, give it a quick workup automatically, and use the crude. So if you had 64 amines and you had one particular acid, you could make a 64 separate amides. They'd have a little bit of amine stock material, a little bit of acid, They might have coupling reagents, some byproducts, some products from the coupling reagent. Try and clean it a little bit, maybe an acid-base wash if the functional groups are not polar, or they're not acidic or too basic and get washed away. And then take those crude individual products. So you've got an individual product in there with other stuff, and see if you can use X-ray on that crude to see if you can grow those fragments. Do any of these vectors lead to enhanced binding? And can we correlate crudely that more binding interactions will equate to better affinity? So that was the kind of the idea. This was funded towards the end of Storm's EPSRC grant. So here's a fragment. We've got a piperazine, we've got a urea, so we've got hydrogen bonds here, hydrogen bond acceptor, furan here. Can we change bits of these molecules independently, grow them in different directions using automated chemistry to try and improve the binding? So we've got one variable here is the urea. Do we need that urea? Do we need those two H's? Do we need this heterocycle? Do we need this right-hand side? Can we chop it down, put a morpholine here? Do we need this furan? Can we substitute this furan with other groups? Can we chop it down? Can we have a urea here? Can we put a methyl group in here to lock it, or to maybe pick up a hydrophobic interaction? So Tobias Kroer, when he was here, put a really crude kind of almost by looking docking, and said, can we make molecules like this using very, very quick chemistry? And it seemed to dock okay by this. And we're talking about lots and lots of years of work to do 4 or 5 different manipulations of this. You might need a team of 6 medicinal chemists for 6 months or even longer full time to put in this work, even longer, and use lots and lots of solvent. Storm was doing the chemistry, Anthony was helping out with crystallography, and then other postdocs and PhDs have worked up on the workflow since, and hopefully his paper's coming out soon. So we can do multi-step reaction in this opentrons, and you can get 20 amines with 20 other amines and get 200 reactions. This was probably an overnight reaction. So you've got a sulfonamide formation, hydrolysis, and you form the acid part of one with different sulfonamides. So you're going to mix those later with this activated urea. So this methyl group enables this imidazole to come off. All these different amines. So you've got 10 different amines. We've got 20 different amines here, deprotect here, and then you put those together. So you've got your 10 different sulfonamide with your acids, 10 different amines here, 20 here, 200 compounds. Imagine getting your PhDs to make those, separate. Obviously, you can purify them, you can characterize them by NMR, get all your data. But it's quite a feat to be able to do this and take it straight to the synchrotron, take it straight to the X-ray, and make your molecule. And that was one of the molecules we're aiming for. We're kind of getting towards that kind of larger, higher carbon content, hydrogen heavy aptan content that you'd expect from a more Lipinski-like drug molecule from a fragment. These are some of the arrays. I won't go through it with too much detail because it's on a chem archives. But let's just look at this particular reaction. Methylate, convert this into a leaving group, have these amines, a couple of different amines, got 116 reactions that we can do crude. We've not had to do any intervention. We're not running NMRs, we're not getting melting points and infrareds. We're making all of these reactions quickly on the synchrotron for synchrotron analysis. What we found was that when we got a particular exit vector, so these ureas are quite useful here. We've got this piperazine here, and we're looking at different right-hand sides, keeping this furan constant. When we had a methyl group on the inside of the piperazine, it acts as a lock. We're able to switch the binding, so they don't go sideways if you want. We call them divers. We change the binding mode in this by making these compounds. And from a couple of months' work, experimental work, several thousand reactions were carried out, a few thousand molecules were made. The mass spec analysis was done using an algorithm that they developed to cut down the bottleneck of checking for the molecular ion. It would find your MS scan automatically. and we found different binding modes. Every time we got a crystal structure and a binding mode, we'd either remake the molecule in-house or get it remade by enamine, so we had sufficient quantity to do some assays with. And we really have a technique really where we can do lots and lots of rapid chemistry automatically, lots and lots of analysis by X-ray crystallography quickly. We got about 29 X-ray structures from this work. We got an active compound that's about 30 micromolar from this work. So we increased affinity by about 6 or 7 fold, and we got this on like an SPR kind of assay from this work. And this is the workflow. We've saved so much time, saved so much workup, so much analysis. We've made crude mixtures, but individual compounds in each well, apart from all the off-target, the byproducts and start materials we had in there. And we've gone from a non-binding molecule against quite a difficult atypical bromodomain. Everyone works on typical bromodomains. We've chosen a pretty difficult target. We got 22 binders. I think some of the binders were just starting materials that we already knew. Developed some SAR and got some binding, and also developed molecules where we knew the absolute stereochemistry of this group. So that was an important finding, that we found out unexpected binding and rich data. We're generating data quicker, saving years and years of effort to get this through. I've come to the end of the talk. Hopefully I've not gone over too long. I hope I've showed you how we can make focused libraries of bioactive compounds. We can make PROTACs, we can make novel linkers, we can target P53, and we can make, it's really interesting from a synthetic point of view. I'd like to thank past and present members of Drug Discovery Centre here. Well, here's Andy McGowan, who I spoke about quite a lot. Here's Claire. Here's Asma, who's worked on some kinase work with us. Mohan's been an excellent crystallographer. Lots of other colleagues here. Got a lot of funding from Worldwide Cancer Research, Blood Cancer UK and OMAIS, CR UK, Royal Society, and EPSRC and these other funders. Thank you very much for your time. And thanks to MCE for inviting me. It's been a real pleasure. And I'd be really happy to answer any questions now.
Okay. Thank you very much, Professor John Spencer, for such a fascinating talk today. We have a few questions in the Q&A sections. The first one from Roland. Have you observed any alene formations after reacting the terminal acetylene with imine? I think that's, yeah.
Yeah, well, I think we did. We've had some side products. What we have seen as well is, that's a very good question, is I'm trying to think what we got, but we had like a cyclization of 3 of the aldehydes to make a 6-membered ring. We have had some unexpected compounds. We didn't look into it much, but the alene is something that we thought we'd got. We looked into it and we couldn't quite fathom it out, but there obviously is this possibility. Yes. I think one of the aldehydes, it cyclizes and makes a 6-membered ring, and you get 3 of the R groups in it. I think the oxygens attack one another. I think it's a 6-membered ring with 3 oxygens in it, and then the R group was the main byproduct that we saw from this reaction. And that's actually reminded me, we ought to put that in the paper that we're writing up, maybe in the ESI. Alene, we've intimated that, we've hypothesized that they were made, but we've never really seen it on a great scale. But a great question. It is something that's conceivable, and maybe something that we could build into some of the reactivity to get that to be the, make some alenes and make them using multi-component reactions. Thank you for that. I hope that answers a question.
Yep. We have another question, more technicals. How do you measure KD, the dissociation constant, with your products?
Well, we don't usually do this. It's usually collaborated, but we can do this in-house. You can do radioligand binding, I guess. So you could have a natural substrate, and you can test it against an antagonist or an agonist and see how it comes off. Or you can get an absolute KD by binding by isothermal titration calorimetry. With P53, for example, a ligand, they'd use ITC, thermal shift, and sometimes they'd use NMR like Waterlocksky. So NMR techniques, calorimetry, radioligand binding sometimes, not so much at the university, but maybe commercially, and maybe NMR techniques sometimes.
Okay. Third question is regarding the P53, the mutant assistant. Have you considered other cis-reactive groups like cyanamides or alpha chloroamide?
Yes, we did try a few, as I showed in an early slide. We just see if I can. I don't know if I'm still in presentation mode. Am I still in presentation mode?
Yes.
Yeah, thank you. We did. I mean, we did test these. I didn't say this. We did test these against, in one of these studies, we tested them against glutathione as well for cross-reactivity. So obviously, you'd have a problem with glutathione and liver tox if you're going to develop these compounds, and you have to rule that out and try and factor that in. We tried mustards, we tried chlorus. One of the problems is this particular pocket, this subsite 3, is quite hydrophobic. We were surprised that Showcap got a CO in there and acrylamide, but there is ample room. I think the problem is actually having the solubility as well. You need fragments that are soluble, and you need a handle in there. So you need some in there. Trying to make these type of compounds or chloroacetamides or other things, and then introducing the nitrogen at the last stage, it's likely to react as well with the side group. And we didn't give it too much time, but we probably need to put that handle somewhere else, not in this subsite 3, because it doesn't sit quite right in this hydrophobic pocket. But obviously there's a whole array, Gunning and other people have written beautiful reviews where they've got reversible, irreversible, thiol reacting groups that can hit this thiol. There are about 13 different ones in P53, especially in Y220C. So trying to get to this one would be really nice if we could template this in there, make this the reversible binding event, and then get this to latch on to, say, a Michael acceptor or another group. We've got to get the resources, we've got to get the people in the group to try and think of how we can do that and get it right. But it is something that's still on our mind. Getting a covalent stabilizer of P53 with good solubility would be really, really interesting.
Right. Thank you. We have 2 questions from Rohan, actually. The first one is, do you have any mechanistic insight as why changing from sulfur to selenium light helped with the changing R1, the blue aromatics, from phenylene to another aromatics? And the second one is, did you explore it at a wide substrate scope for the copper catalyzed multi components reactions, such as alkenes instead of alkenes, and replacing the pyridines with other heterocyclic nucleophilic?
Good. I'll just do the second question first. I don't know if this reaction works. It's probably a question of acidity of that alkyne. To get the vinylic group to react would be quite difficult, but I guess you could do a kind of a hydroboration and then put copper in there, but I'm not aware of this A3 reaction working with a vinyl. It's a very good point. Maybe we can hydrogenate the triple bond down to totally saturate it. Maybe we could use something like a Lindlar. So we could make this, and then we could make the vinyl from this, or make the. That's probably the way of doing that. We've hydrogenated it totally down to the alkane here. We can do that. Jan's done that. Jan Lin in my group, she's done that by hydrogenating this on an H cube. The vinyl, I don't know about the chemistry. It's almost feels like a Heck reaction, almost like some kind of Heck type chemistry with palladium. We haven't done that with copper, but it's a very good point. Maybe we could use a kind of a Lindlar like poison catalyst, or something like a diamide kind of I think you can do a birch reduction or a modern variant of that to get the double bond there. But we haven't actually tried that. Didn't quite. Could you read out the first question, please? It was to do with the selena sols, wasn't it? To do with the selenium.
Yes. It's regarding the Hansch thiosynthesis. Do you have any like a mechanistic insight as to why changing from the sulfur to selenium like helped with the changing R1 from phenyl to another aromatics?
Okay. Well, I'd expect selenium to be more nucleophilic and more reactive, but we didn't really pursue this. Selenium is horrible smell. Sulfur's bad enough. Working with sulfur, I think selenium molecules are pretty smelly. So we kept this kind of light touch and just made a few analogs and did this. This might be something for Storm to consider to work on this. It's probably also to do with the availability of the starting materials as well. So I think we just wanted to show selenium works. It works quite good. I'm sure a lot of the chemistry we can do. Storm's actually developed this even further now. He's got a metal-free route, but I won't go into details because it's his chemistry, it's his independent chemistry. Now he's at Sussex, and they've shown it works with selenium as well. So selenium will work. It's probably likely to work. They're quite rare. One of the referees for All Black, they were very supportive, said it was quite nice because there's not many of these selenosols. So it's maybe something to exploit, but somewhere where you're maybe in a lab where people can't always smell it, because working with these kind of chemicals is bad enough when you're working with sulfur and people keep thinking there's gas leak because they put sulfur material in gas so you can detect it. They put thiolate, methyl thiol in gas so you can detect it, and it can be problematic in a laboratory environment. So we haven't taken the selenium chemistry as far as we would have. We just wanted a light touch just to show you can make them. And if anybody wants to make them, just make use of our chemistry. It should be useful. But I think availability of selenium, it's probably going to be more reactive, more nucleophilic from a mechanistic point of view. The availability and kind of the handling of it precluded us from taking this even further. Storm's PhD student, Joe, is doing really well. Joe Smythe, really super PhD student, might want to make more selenium, but he might want to put a peg on his nose and maybe do it when other people aren't in the lab a lot on a very windy day with all the windows open. Yeah, I think that's probably the answer.
Okay, great.
Nice to hear from you, Rowan. He worked with us recently. Yeah.
Okay. Yes, I have one question, very quick questions. So for your plug and play PROTAC molecules, and did you just, am I understanding right? Did you just create a three-way adapters? You can link like three moieties together. That would be quite exciting. You can put like a fluorophore or maybe a targeting moieties onto the
Jan's made that. Well, we haven't gone and made the PROTAC, but Jan Lin's got we've got a Bo-Dippy, we've got an E3 ligase, and we've got an alkyne, and we've got a handle on the other end. It's almost like choose your protein of interest ligand. As I said, we're not in the game where we have a particular, we're kind of disease agnostic. We're not thinking of JQ1 or whatever kind of ligand on there. Gives the people the opportunity, but there's an opportunity from that chemistry to have different linkers, different fluoresceins, Bo-Dippies, Dansal groups, whatever you want on one part, an E3 ligase ligand such as maybe a PEG or an alkyl, where you can have an amide bond or an alkyl bond, a clickable PROTAC, VHL, decaf, IAP, or MDM2 ligand on the other end. So there's the actual the actual potential is limitless. You can make 1,000 analogs if you want, different lengths, different properties. We just made one. We just wanted to show this A3 chemistry can be used. Here's some examples. Here's the JQ1 examples. They've got activity, and we've definitely got. Let me just go to illustrate this better. We've got an example where we've probably got on this side, we've got a nitrogen linker here going out to a Bo-Dippy, with some carbons in it. We've got a E3 ligase ligand on this part, and on this part, we've probably got a bromo handle or an OH. So you can put whatever protein of interest you want here. And then here you've already got your BODIPI, and here you've got your E3 ligase. So just clip it on to your protein of interest, stick it into cells, and you might have a way of watching degradation in cells by microscopy or something. It might even be better to keep this handle free, just have the E3 ligand, and people can put in whatever fluorophore or whatever cell marker they want, and then whatever they are. It's just a matter of funding, having the funding to take this part through, having the personnel to do it. We just wanted to show the potential of this chemistry without, you know, cranking the handle and making 1,000 analogs if we didn't have the time. So we've kind of done what you've said. We haven't made a PROTAC, but we've put the elements on. There's a BODIPI and there's an E3 ligase ligand. Someone can get that and put on there. It might not be the right length for the E3 ligase. The BODIPI might be too short. It might not have the right hydrophobicity. Like a lot of these things, you'll have to, it's empirical. You have to make quite a few analogs to get it right.
Yes.
But at least we've got the kind of chemistry there where someone can quite quickly do that. And why not have a BODIPI on here, have an E3 ligase on here, and have your ligand here, and do it quickly? You could probably assemble these molecules. It's Lego chemistry, isn't it? You've got some building blocks, just put them together. In one step, we should be able to do that chemistry. And that's the kind of thing that we're looking for to do now, to try and do that and have a particular POI to target a particular protein in cancer. And that should become feasible using this chemistry. Why go to the project? You know, if you at MCE have got 100 different ones of these with different cell markers, you've got 30 or 40 of these with different E3 ligases on there, and you've got lots of alkynes with drugs. You could quickly make lots and lots of these molecules, and I'm sure some of those would have biological activity. It's just having that kind of feedback that we don't quite have the resources to do at the moment. It's quite interesting though that making chemistry, you're making more complicated molecules, doing more complicated chemistry, but much easier and much quicker, and that's what's quite nice. We're expanding our kind of opportunities and our kind of what's the word, complexity and 3D nature, but we're doing it with really easy chemistry. It's almost like anyone can do this, almost like bucket chemistry. It could be an easy thing, almost buy a pack, mix them together and make the molecule. So biology group could one day maybe buy the components and mix them and test them and do them to order. So it's quite readily, you know, open-tronable chemistry that we hope to be able to do. You can do it on a robot and do it quite easily. Okay.
Thanks a lot again, Professor Spencer, for such a great talk today.
Thank you.
And thank you all the audience for coming today for this webinar. There will be more exciting MCE webinar coming soon in this November. Our next speakers have a medical background, and we will be discussing a different aspect of the drug discovery, focusing on the association between the genotype and the drug response. So please stay in tune. At Medical Express, we are committed to fostering a scientific communication and accelerating the pace of scientific discovery. So thank you again for joining us today, and stay connected. And I am looking forward to seeing you all very soon. And goodbye now.
Thank you very much. Bye-bye. Thanks.